Yeah. Thank you, everyone. Thank you, everyone, for joining in. Today, we have Dr. Johanna. Johanna is a staff scientist in the Material Science Division at SLAC Laboratory. Her research centers on X-ray microscopy of materials under realistic conditions. Dr. Nelson is an established researcher in this area of energy storage system. I welcome Dr. Johanna, and now I would request her to go ahead with the presentation. Thank you, Doctor.
Thank you. Thank you for the introduction. So I'm going to talk today about X-ray microscopy of operating electrochemical energy storage systems. As was introduced, my name is Johanna Nelson Weker. I work at SLAC National Accelerator Laboratory, so we actually pronounce the acronym, and so we call ourselves SLACers sometimes. And I work at the X-ray light source, one of the two X-ray light sources. So let's get started. So because I wasn't sure of everyone's background and how much expertise people had or knowledge about SLAC, I wanted to give a small introduction of the laboratory, look at different X-ray sources that we have there, and then specifically dive into X-ray microscopy. It's not the only technique that my group uses, but it is the primary technique that my group uses. And then after that, I will go into two main topics, using tomography or 3D imaging and 2D imaging to look at morphology changes, volume changes, and density changes in two sort of systems for alloying anodes. So a germanium anode, which is sort of analogous to silicon, and then a tin anode where we've done nanoporosity to see if we can mitigate these density and volume changes. And then the second example is combining X-ray microscopy with spectroscopy so that we can look at chemical changes. And this we can do 2D images of what's happening chemically in the battery. And I have two examples of that, lithium cobalt oxide cathode just as a standard test example, and then a conversion anode using nickel oxide. So SLAC National Accelerator Laboratory, since I expected the audience to be somewhat global, I put us on the map of the United States. So we are sitting, let me get my pointer, here in California. It's 6:30 in the morning, so it's still dark out. But when the sun rises, we are sitting right next to the San Francisco Bay. Stanford University, we are operated by Stanford University, which we have a lot of Stanford students coming and postdocs coming up. They sit here. You can see Hoover Tower, and all the red roofed buildings are all Stanford University. And then about 2 miles or 3.5 kilometers away is the campus of SLAC. The main campus is sitting here. And we do have a 3+ kilometer long linear accelerator that used to do high energy physics. It's now converted to do photon science. And I'll talk a little bit more about that. So we are funded by the Department of Energy, the US Department of Energy, and we are operated by Stanford University. So we have two X-ray sources on site. We have a free electron laser and we have a synchrotron. And the difference between them is the quality of the X-rays that are coming off. So if you look at peak brightness, which is essentially the number of photons you get, it is per second per area at some angle and some bandwidth. So if you look at the peak brightness, our linear accelerator has been converted to the LINAC coherent light source, or LCLS, and that is very laser-like light. So it is coherent like a laser, single energy, and in small bunches. And so you can do, because of the high peak brightness, of a single bunch, you can do very fast time resolve measurements. However, if you look at the average brightness, it's actually not as good as the synchrotron.
Mm-hmm.
They're actually coming up with a upgrade. This is a little older of a plot. So the upgrade will have both a high peak brightness and high average brightness as well. So down here on this log scale are a bunch of synchrotrons that are around the world. And so we sit down here with the synchrotrons at Stanford Synchrotron Radiation Light Source, or SSRL. We are originally a generation 2 light source, but we were upgraded to a generation 3 light source in the early 2000s. And so we are also looking always for another upgrade if we can convince the US government to provide us another upgrade. We started out as a high energy facility, SLAC in general, and both of these light sources started out doing high energy experiments, energy physics experiments. So we actually have a collider hall next to our synchrotron where proton and electron beams used to collide. But now we're completely gutted and redone as a dedicated photon source. So we use X-rays to characterize materials. We also have a large biology section as well, but my research has been focused on materials. Little more specifics of the ring are given here. I'm not going to go through them, but we do have a bunch of US and international users. So we have almost 2,000 users a year. And to get beam time on one of our instruments, you just submit a proposal. It's freely available to anyone whose proposal is peer reviewed and scored well. So we have 32 experimental stations, but we're also constantly building more. We can do X-ray scattering, spectroscopy, and imaging and microscopy, and they're scattered around the ring, different techniques. Every beam line is really honed in on a specific type of experiment to do something very unique. And you can always take your one sample and bring it to many different instruments around the ring and really get a wealth of information about one single sample by using multiple techniques. And that's really what my group research focuses on. And we focus on looking at batteries. And the beauty of using X-rays to study batteries is that you could do this operando. You could do it while the battery is operating.
Good.
So batteries, yeah.
Do you want to take questions in between or at the end?
Either way.
Yeah.
I think
I just
in
have one
between is
question.
fine as long as it's not, yeah.
Yeah. For the international, how does that work? Do they do remotely? How do the proposals work? If you
Right
have
now?
to use a facility. Yeah, now I think everything is.
So everything's remote right now. International users usually we have them come on site in normal times. Because of the COVID virus, we are trying to put as much of our instruments remote and remote access as possible so that users can remotely take data from their home country. But that's obviously something that's going to take a while. There are some experiments that are more readily available than others. So for example, some of our scattering beam lines, we now have robots for the, sample changing.
Got it. Good.
Yeah. So X-rays, and we're a hard X-ray facility, which means we are higher energy, give us the ability to look inside batteries. And batteries are really interesting as a system to look at operando because, as you all know, they're heterogeneous, they're hierarchical in the dynamics. There's things that are occurring at all length scales. For example, it's really interesting to look at the atomic scale with scattering and spectroscopy to know what's chemically or structurally changing in a battery. It's also interesting to look at the nanometer scale where you have the primary particles and what are they doing over time, and the chemistry and the morphology changes that are happening there. But at the largest length you really also want to look at the micron and even the millimeter scales of what's looking at a more realistic battery or a larger field of view of what's going on in your battery. So you really want to study it at all different length scales, and you really want to study it as it's operating and changing, and most realistic battery as possible. And so X-ray characterization allows us to do this. We can extract the chemistry, the structure, the morphology, and all of those changes while cycling a fairly realistic battery. So that's the aim. And this puts it in a slightly different way, more focused on what my research does. So we are able to look at batteries as full cells. This is an 18650 cell that we looked at, not at SSRL because our energy is just a little too low for that, but we went to another synchrotron, the advanced photon source, and took data there. We routinely look at half cells, and all of the data I'm going to show you today is mostly in this area here, looking at half cells, looking at specifically one electrode, either the anode or the cathode, to see changes over time, chemical changes and morphology changes. We can look at the particle level by zooming in even more, doing a little higher resolution. This is a tychography example that we did. This is not a battery. This is a catalyst sample, but it gives you an idea. We can get down to about 5 nanometer resolution. And then we also look at the atomic level. This is XANES, and I'll show this data later, but we can look at bond lengths in crystalline and non-crystalline materials. And then finally, and I touched on this a tiny bit, because we're at a synchrotron, we can actually change our X-ray energy. And so we can tune our X-ray energy to look at different chemistries. So synchrotron, the SSRL synchrotron sits here in the hard X-rays, so at higher energy, smaller wavelengths. So we can look at transition metals, the K edges of them, or even going up to platinum, L edges, M edges. If you go down in energy, you can actually look at things like sulfur or magnesium. And we have a few beam lines that do that. And we have also a few beam lines that go down to the tender or soft X-rays, where you can look at carbon or oxygen or fluorine. And so all of these chemistries are really important in batteries. And so the ability to choose your energy, depending on what you want to look at, is a big benefit of having a synchrotron. So today, though, we're going to focus on microscopy. So specifically, beamline 6.2, the transmission X-ray microscope, or TXM. So I call it a microscope because it does have optics. This is what it looks like in schematic form. It's very similar to a TEM, a transmission electron microscope, and that's why we named it TXM, because of the similarities. But because we have X-rays, we can actually penetrate much deeper into samples than you would with an electron. So the details aren't really that important. X-rays are going from the left to the right. So everything on the left is just giving us the nice beam that we want, energy tunable, monochromatic X-ray source. And then here's the actual microscope. And it's really, you can simplify it by doing just lens drawing. So we have a capillary lens that focuses the light, defines the field of view for our microscope. We have the sample stage. We can do X, Y, Z, and rotation. And then we have the most important part of a microscope is the objective lens. And that really takes the scattered light from our sample and creates a real image. And that creates an image on the scintillator, and we actually convert our hard X-rays to visible light, so we can use a CCD camera, which is just faster and a smaller pixels. So that's how we image. And the features of this microscope are such. Because it's full field microscope, we're not scanning the sample. We can take images in half a second, and we can bump that up. We're not photon limited. We're pretty much detector limited at this point. We have 30 nanometer resolution, and that's from our zone plate optics. We also have a 30 micron field of view. These are both dependent on the energy a little bit because we can tune our energies. Those change a little bit. But here you can see we do mosaic mode imaging often. If we have a sample that's larger than our field of view, we take multiple images that are overlapping and stitch them together to make a nice continuous field of view. Because it's high X-ray energy, we do focus on in situ samples, and I'll go into that in a little bit. And then we can tune our X-ray energy to look at elemental or chemical mapping. And the energies that we have access to is from about 5 keV to 13 keV. And these are the Z numbers on a periodic table that we can then focus on because of those energies. So the absorption of a photon is determined by the number of electrons. So the Z number matters a lot. And then finally, we can do tomography or 3D imaging. So this is what the actual microscope looks like. Unlike an electron microscope or an optical microscope, X-rays are coming out from the side, so our microscope is sitting planar rather than up and down. And like I said, we focus on in situ experiments. So we have a lot of different in situ environments that we can do, not just batteries. So over here on the right is a heater that we do catalyst samples on. There's a little nanoreactor that can flow gases at certain pressures that goes inside that heater. So you can look at materials as you change the gas, the heat, so the temperature and the pressure. We have a diamond anvil cell. That's a top-down view of the diamond anvil cell, so we can do very high pressures. We do batteries, and then we also have a 3-electrode open cell for aqueous solutions, which also has a small heater and some bubbling if you'd like, some oxygen bubbling, things like that. And we routinely design or help users design whatever they need for their specific technique. So this is not all that we have, but these are the ones that we most often use. So often we'll build something with a user, but keep it at the beamline so other users can use it then. Obviously after the first user publishes. So that was the outline of the introduction of X-rays and things like that. So next I really want to go into these alloying anodes. How do we track morphology if we don't care about chemistry, but we care more about morphology changes, volume changes? Before I do that, I want to show you sort of what we now do for our in situ cell. So it's taken us a number of years, about 10 years, probably 5 years, to perfect electrochemical cycling at the beamline and make it so that it's indistinguishable from what we do in the lab. So we always use pouch cells. We use aluminized pouch cells unless we're in the tender X-ray regime, then we have to do modifications. But for everything you'll see here, we use aluminized pouch cells. We sandwich those pouch cells sideways between 2 aluminum plates. And we have an imaging hole here. And rather than keep that hole open, we do put beryllium windows on the inside of the aluminum outside the plate, outside the pouch. So it puts constant pressure across the whole battery. We found that gives us the most consistent and long-term cycling. So we are able to cycle for tens of cycles. I don't think we've ever cycled hundreds of cycles just because we ran out of time. Yeah.
And Johanna, in this context, because some of us are power electronics engineer, what does in situ mean in this context?
Yeah, so I'm using in situ very broadly because it definitely depends on the field. I would say the battery field, in situ is not well defined. We are almost always cycling during data collection. And I'll specify that depending on the material. So in situ, I would say it's sort of broad. I try to use operando if it's really operating. We use a mix of in situ, ex situ, operando, depending on what's best for the amount of time you have and the questions you want to answer.
So in this case, it means the battery that you're cycling, that's it. In this context.
Yes. Yes. Yeah. For catalysts, in situ usually means you're putting it into operating environments, but you're not measuring the performance.
Got it, got it.
But for batteries, it's, I mean, you measure the performance when you cycle it, essentially. But yeah, this is standard cycling.
Got it.
Okay, so I promised the first example was going to be germanium. These alloying anodes, they're high capacity, but they have a very short lifetime. And the reason for their short lifetime is to accommodate this large capacity in these metals. As you alloy with lithium ions, the volume changes dramatically. So in silicon, you can get like 300, 400% volume changes. That obviously over time causes cracking, fracturing, an unstable solid electrolyte interface, which can grow continuously and eat into your lithium reservoir, and it severely limits your battery lifetime. But because the capacity is so much higher than graphite, they're still really interesting to look at. And silicon has been put into commercial batteries, although maybe not as successfully as everyone was hoping. So we chose to look at germanium, and this is a fairly old paper, and I just included this because it's the best tomography paper that I have. We chose germanium because it was going to be the easiest thing to see, because we could go to the germanium absorption edge for x-rays. Silicon was going to be much harder to see. We did some follow-up studies on silicon after this, but in terms of image quality, this is probably the nicest. So here you're looking at a 2D black and white image. It's just an image of the absorption. So bright are the germanium particles. They absorb the most. This is in a binder carbon matrix, but you can't see it. The x-rays are insensitive to all that material. And so what you're going to see once I start this video is as we lithiate, we're going to form the largest particles are going to start to crack. The cracks do fill in as you continue to lithiate. So when it's fully lithiated, it looks kind of fluffy, but fully sort of no cracks. And then the cracks do reappear when you delithiate. So I'll start the movie. Nothing happens really until we reach this bend, this just SEI growth. And then you start to see cracks forming, and then they fill in as you continue to lithiate. And then as you delithiate, you do see those cracks reappearing. They're porous particles, and the particles don't return back to their original 2D area. But that's just 2D. We really wanted to push the technique, demonstrate that we could do 3D imaging while we were imaging the battery. So 2D imaging can be misleading. Sometimes you need an extra angle to figure out what's really going on. So the way that 3D imaging is done in the full field microscope is tomography. It's basically a CAT scan. It's computed tomography. So we take images at many different angles of our sample. Unlike medical CT, we rotate our sample. You don't want to rotate your patient, and it's really hard to rotate a synchrotron. So we use the same computer algorithms essentially and convert your 2D projection images at different angles to a 3D volume of XYZ. And then you can take that volume and really rotate it, slice it, and dice it any way you want to see what's inside. Because of radiation damage, we weren't able to do this truly in situ or operando. And so we did it maybe quasi in situ, where we looked at these two fresh particles here. We lithiated without x-rays, looked at them fully lithiated, delithiated without x-rays, and then looked at them fully delithiated. So these are the same field of view, but we don't have snapshots in between, just because the radiation dose of taking tomography continuously was just too much. But because they are 3D, you can rotate them. And for the first time, you can actually see that delithiated, these aren't just cracks. They are completely severed particles. And ideally, that's getting bad. So I'll just continue. But ideally, we would also be able to look at them in between to see when exactly the cracks are starting to form. But because we have 3D volume, we can also get the exact measurement of the expansion. So we did calculate that there's a 320% expansion. And it's hard to see right now, but you can also quantify the density because we know that we start with pure germanium. So we did that here. We basically took sub-volumes inside the particle and made sure that they were completely inside the particle and a volume outside the particle for normalization. And we wanted to do this not using the whole volume, just to make sure that we were avoiding edge effects of looking at the edge of the particle. And we plotted the number of voxels and the germanium density. We assumed the germanium density was pure germanium at the beginning. And so here is just centered. We forced this red line to be centered around the germanium density, and then we could see the changes in the germanium density, assuming no lithium. So we're insensitive to lithium. So if we're creating pores or if we're adding lithium, we're reducing the density. So we can assume as we're adding lithium, this dashed line here is what we would get if we got lithium 15 germanium 4 fully lithiated. So we get near there, but definitely not centered on there. So we have some lithium 15 germanium 4 mixed with other lower lithium content states. And then when we delithiate, that's the blue curve, you can see it's broadened out. There's a large variation between the densities, and it's shifted to lower densities compared to the pure germanium. So we're either leaving lithium in there or leaving pores in there. So it's not completely reversible. And then we have a follow-up paper led by Linda Lim, which I would suggest you look at, which uses diffraction and spectroscopy to really confirm what those species are. So with the combined technique, we're able to get the real full story. So that was a fairly old paper, so I wanted to look at more, show you what we're doing now as a follow-up, and we're looking at tin. Tin is also a good alloying anode. It's maybe more easily to get out of the ground than germanium, but it suffers from the same problem. And so to mitigate this problem, we can think about trying to introduce porosity ahead of time. So we have collaborators from UCLA, and they've looked at doing nanoporosity to extend the battery life, and they've done that very simply by doing selective dealloying. It's a very easy technique that's not hard to scale up. And that gave them nanoporosity of about 25% internal porosity with pores that are on the resolution of what we can see with the TXM. So then they came to us and said, hey, we want to look at these. And you can see, if you look at the nanoporous tin, it's very stable cycling over the 200 cycles compared to dense tin, which really just didn't cycle at all. So this is what the tin looks like, the nanoporous tin looks like in the transmission X-ray microscope. You can see, especially on the edge of the particles, there's very easy to see single pores. And we wanted to see how these pores change while cycling. So we looked at...
Yeah, when you say cycling, at what C rate do you typically cycle the cell?
We try to go at least C/5, C/10, C/8 is usually what we're trying to aim for. If we're running out of time, we go to C/3, but we want to stay away from really fast cycling, unless that's the point.
Got it. Thanks.
Mm-hmm. Yeah, I'd have to remind myself by going back exactly what the C rate was for these cells. I think these were C/5.
Yeah, good.
Yeah, so the first row is a dense particle of tin. The second row is the nanoporous tin. This is during lithiation. I've circled the outside of the particles just to give you an idea, better visual of what's happening with the shape. And you can see the dense tin doesn't change at all until the very end. And that's actually very similar to what we saw in the germanium. We weren't seeing much change at the beginning, and then suddenly cracks start to form. And so then there's this burst expansion near the end. It explodes into something much larger, and cracks form. The nanoporous tin, on the other hand, doesn't really change area very much, but it does change shape. And we were sort of disappointed by that. We didn't expect it. And so if you plot the areal expansion as a percentage, black curve during lithiation is the dense tin, whereas the green curve has a small areal expansion, but it's much, much smaller. During delithiation of the same two particles, you see that the dense tin doesn't return to its original shape, doesn't return to its original areal expansion as well. So it's significantly less dense. Whereas the nanoporous tin, if you track the area, does return to its original area, although the shape has changed. And if you zoom in, the pores have changed as well. And I also want to mention that these aren't the only two particles we looked at. We tend to look at like 15 different areas within the battery as we're cycling the battery. So these are just representative areas. So porous tin didn't work out as well as we wanted it to. So we brainstormed with our collaborators from UCLA, and they suggested, well, let's look at an intermetallic. And okay, maybe putting antimony in a real cell isn't the best way to do it, but we can at least observe what's happening. Can we make a more stable structure by having an intermetallic where both of the metals will alloy, but they'll alloy at different potentials? And so one will be the structure while the other one is alloying, and vice versa. So we looked at tin antimony alloys. And so here we're comparing on top this nanoporous tin again, and on the bottom is the nanoporous tin antimony alloy. And so these are just snapshots. We took more images than I'm showing. These are snapshots. This is before cycling, fully lithiated and delithiated. Again, the nanoporous tin is changing shape as we had before. The nanoporous tin antimony actually cracked, which we were a little surprised with. But it does return to its almost same area as before, without much structural damage. So if you plot the areal expansion again, the gray is the nanoporous tin, the blue is that bulk tin that we saw before, and it's the same data. We just replotted it. And then the brown is this new data of the nanoporous tin antimony, and you can see it expands as much as the nanoporous tin, but during delithiation, it does go down. And if you look more closely at the pore structure, it seems the pore structure is more stable. So these is just a small field of view of the larger field of view, zoomed in, essentially. It's mostly just cropped, it's not zoomed in. This is the pore structure before cycling. This lithiated, you can see the pores have gotten really large. Yellow is the tin, black is the pore. And then delithiated, the pores have actually agglomerated, the pore walls have fallen apart and things like that. Whereas the nanoporous tin antimony, it seems the pore size is actually maybe even smaller to start out with, and maybe that helps. There's not much change as you cycle it, as you lithiate it and delithiate it. So we plot the average pore area, and you can see that really this is pretty stable with lithiation and then delithiation. There's a small expansion, the pores get filled in, and then there's a small contraction as the pores reform, essentially. But it's a lot more stable than you see in the nanoporous tin. So we think it's actually the pores and not necessarily the general shape that's really helping us with this nanoporous tin antimony alloy. Okay, in the remaining of the talk, I would like to look at chemical imaging because I talked a lot about being able to tune the X-ray energy, and we can then use that to combine spectroscopy with microscopy.
Yeah, Johanna, before we go to the next, can we take quick question related to porosity?
Quick.
Yeah.
Definitely.
There is one question from Murat. I hope I'm pronouncing the name properly. Have you considered different porosity levels for comparison apart from 25%?
No, we haven't. Yeah, that's a great question. We haven't explored that in this work, at least. But that would be, yeah, that would be useful to do.
Yeah, please go ahead. We'll come back again.
Yep. So lithium cobalt oxide is probably not the most interesting thing to explore. If you know anything about batteries, you know that we can reversibly cycle half a lithium in and out of lithium cobalt oxide. And it's because of the very stable layered structure that it has. But if you take out more than one lithium, or more than half a lithium, for example, the structure does start to break apart. You can see that stability if you look at the X-ray absorption near edge structure of cobalt. So here, this is the XANES of the cobalt edge of lithium cobalt oxide in a operating battery. So this is, red is OCV, open circuit voltage, blue is fully charged, that half lithium. And then the dashed black line, and I had to make it dashed so you could actually see the red line, is fully discharged. And so you can see it's completely reversible. And this is the first cycle even. So it's a very stable structure. But we wanted to explore what happens when you over-lithiate it. So if you have a number of batteries in series, which is often the case in electric vehicles, you're really not measuring what's happening in each individual battery. You're looking at the stack of batteries. And so it's possible that a single battery might be operating slightly differently, and there's a chance of over-lithiation. And oftentimes, those fires and accidents and things like that are just because of one battery not behaving like the rest of the batteries. So over-lithiation occurs in lithium cobalt oxide, and when you can put into up to 3 lithium atoms, and you should form cobalt metal and Li2O. So we wanted to explore that with X-ray spectroscopy and then combine it with microscopy. So just in bulk, looking at what's spectroscopically happening during cycling, when we discharge to 0.8 volts, for reference, this black curve is lithium cobalt oxide. The red curve over here is cobalt metal. And so what we see in time is that the white line, this bright peak, decreases as you're going closer and closer to cobalt metal, and the edge shifts to lower energy, indicating a lower oxidation state. Now, because we have these reference spectra, and we know them very well, we also have an idea of what chemistries to expect. We can do linear combination fitting and fit all of these spectra to either lithium cobalt oxide or cobalt metal, or some weighting between them. So we've done this. This is the electrochemistry of putting in those 3 extra lithium ions, sorry. And we start out, this is the percentage of black squares are lithium cobalt oxide. The triangles are cobalt metal. We start out with 100% lithium cobalt oxide, get to about 13% at the end of that cycle, and equivalently replace that with the amount of cobalt metal. And so we get about an 87% conversion to cobalt metal. That agrees well with the electrochemistry, which said we got about 2.6 lithium ions stuffed inside. And we also looked at other cobalt oxides that could possibly be forming, but for example, cobalt oxide does not, COO does not improve the fit, and it just fits the noise. So we're pretty confident that we only have lithium cobalt oxide and cobalt metal. But I'm a microscopist. I want to see this. I want to have the benefit of having the morphology change and the chemistry change at the same time. So the way we do that is, oh, sorry, I jumped ahead. We did also take XFs because we get that for free. So we looked at the extended structure, and that also suggests that we're forming nano cobalt. So we have a cobalt oxygen bond here, a cobalt cobalt bond here. They both decrease as you're reducing the lithium cobalt oxide, and a third peak for cobalt metal starts to grow in. It's not nearly as strong as you'd see in cobalt metal foil, and that's because it's disordered. So it's nano structured. We also did Operando XRD, which I'm not going to show, and that agrees as well. So we can see that the lithium cobalt oxide peaks are decreasing. It agrees with this 13% remaining. And we do see some amorphous or nanocrystalline LiCoO peaks growing in, and a little hint of cobalt forming, but it's nanocrystalline, so it's hard to see with diffraction. So we're very confident that we can see cobalt and lithium cobalt oxides. And those are the only two species that we have. So now we're going to do imaging, sorry. Jumped ahead. So yeah, I promised we could do chemical mapping. So how do we do that? To do that, we take an image at a specific energy. We change the energy, take another image, and keep changing energies. And if you line those images up, each pixel becomes a spectrum. And similar to how I did linear combination fitting on the single spectra, as I did in the bulk sample, we can do this for every single sample or every single pixel on our detector. And so we've done this. These are two different batteries, two different locations during over-lithiation. Red is lithium cobalt oxide, green is cobalt metal. You can see that the shell starts to convert first, which is pretty logical. So the green is forming from the outside in, the green forms, and then particles start to fracture. And then finally, once the particles are fracturing, the core can also convert. So one thing to note is the transformation, the chemical transformation. These are the first three shots. Does lag behind what we see in bulk X-ray absorption. And we think that's due to the chemical limit, the detection limit of our imaging technique. So we're not nearly as sensitive as you would be in bulk spectroscopy. But we do see that the fracturing happens after the conversion starts. So we start to convert cobalt, then particles fracture. So a small over-lithiation is probably okay, but you don't want to go deeper so that you're fracturing the particles. So now that we know that technique, I also want to apply it to a second technique, and this is the last example I have, and it's a nickel oxide conversion anode. So conversion anodes are a third type of anode. They are also have high theoretical capacity, and they also have a large morphology change in that first cycle, similar to alloying anodes. We also have the additional problem that they have low electronic and conductivity, which gives them large voltage hysteresis. So this has been overcome in the literature by nanostructuring. So there's lots of examples that you can see in the literature of different nanostructuring that people have done with different degrees of success. So we wanted to look at both changing the nanostructuring and also the chemistry. So a visiting scientist in my group synthesized this porous flower-like structure of what we later discovered was a nickel oxide hybrid. It was not pure nickel oxide. It has 10 nanometer pores. You can see the SEM images, they're beautiful. There's TEM images, you can actually start to see the pore structure. It shows that it is crystalline nickel oxide, but if we did spectroscopy, we found that it was not pure nickel oxide, but as a small percentage of nickel hydroxide, which is the precursor material. And so we looked at the cycling performance of the nanoflowers with this hybrid nickel oxide, nickel hydroxide structure, a commercial nickel oxide powder, and then nanoflowers that we synthesized further so that we could get rid of all of the precursor material. So they were pure nickel oxide. And we found that this hybrid structure actually cycled much better. So it wasn't just the nanoflowering structure that allowed this long-term cycling, but something else, something with having a little bit of a precursor in there allowed it to cycle better. So we wanted to investigate that. So the first thing we've done is we looked at the transmission X-ray microscope, and this is done ex situ this time, just because of the amount of time we had. We didn't do in situ, but we are able to see, this is just 2D imaging, black and white, of the commercial nickel oxide on the bottom and the nano-flowered hybrid on the top. And we're looking at 4 different locations: pristine, first lithiation, partially through the third lithiation, and then the third, sorry, delithiation, and then the third lithiation. And you can see even after the first lithiation, the commercial structure falls apart completely. It's a little harder to see with the nanoflowers. The resolution of the TXM isn't as ideal as you'd like to be able to see the petal structure very well, but it does, we've looked over many, it does maintain its structure. It does get a little fluffier. That's a technical term. And then those 4 fields of view, we also looked at the chemistry at those. And you can see the chemistry actually behaves very similarly. It's pure nickel oxide, and we had to take out that 7% of nickel hydroxide for these analysis, because the TXM, again, the chemical sensitivity of the TXM is not as high as a bulk XAS. So we just looked at nickel oxide in green and nickel in metal in red. So the pristine shows both are nickel oxide. The first lithiation, they're completely nickel metal. The partial lithiation, though, is a little different. And so we explored that a little closer. So if you look on the right is the commercial nickel oxide, on the left is the Nanoflower hybrid. And you can see the hybrid is more of a brown color. And if we looked at specific spots, this is I and II, they both fall pretty squarely in between nickel oxide and nickel metal. Whereas the commercial structure was either red or green. There was no mixing. So there's very few brown regions, mostly red with spots of green. And so if you look at III or IV, they're either almost completely nickel oxide or almost completely nickel metal when you do the linear combination of that specific pixel. So this suggests that the hybrid nanoflower is more connected, well connected than the commercial powder, and then has more faster kinetics. So it's lithiating and delithiating more uniformly because of the structure that it has.
So do you get a black and white image and then you overlay the color on it, or how do you color code it?
Yeah, so I color code it using linear combination fitting. So the colors are just a percentage of the percentage of nickel versus nickel oxide. So if I don't know the chemistries, if I have no idea what species of nickel to expect, I can look at the oxidation state change by looking at the edge of each of the pixels. You can plot that as a color, or you can do something more sophisticated like some cluster analysis or something like that to be able to create clusters that are pixels that are very similar. But this is 2 million spectra, so you can't look at every single pixel by itself.
Understood.
Okay, and then a little more electrochemistry just to push this a little further on what exactly is happening. We looked at the GITT and found that there's a much lower over potential for the nanoflowered hybrid than the other 2 systems, the commercial, which isn't surprising. That agrees with what we see in the chemistry of the TXM, and even the nanoflower, that's just the nickel oxide. So in summary, the nanoflower structure plus the presence of a little bit of the precursor ends in good performance. It's got higher capacity, it's stable over at least 10 cycles, what we cycled. It's got stable morphology and lower over potential. And also we did higher rate studies, and we can see that it also cycles better at higher rates compared to the nanoflowered structure of just nickel oxide and the commercial material. So using a combination of X-ray techniques and electrochemical techniques, we're really able to explore why this material is so much better than the other two. And then just to wrap up, because it's been a long time, I want to remind you of sort of the broader picture of using X-rays to characterize batteries across different length scales and across different techniques. We focused a lot here on these half cells, looking at chemical changes, but my group does do lots of other techniques as well, depending on the question, depends on the tool we use. And then also we can access different chemistries using different X-rays. The harder the X-rays, the easier it is to do realistic batteries because the penetration depth of the hard X-rays is much larger. And then finally, this is not my research, this is my group's research. So thanks to my research group, we don't have a recent group picture for obvious reasons. So these are my current research group members. The work that I showed you, the nanoporous tin antimony work, was done by Jesse Coe, who's now the staff at Johns Hopkins. And the nickel oxide work was done by Yang Ren. He's a visiting scholar. And thank you. I'll take any other questions.
Yeah, yes. Thank you very much, Johanna. We have lots of questions. We'll just zip through them quickly. Gustavo has a question. How do you ensure that you're tracking the same particle during charge and discharge cycles? Do you have to continuously see the particle or observe the particle during the operation?
So for example, the germanium work where we just looked at pure, pristine lithiated and delithiated, we didn't look at it continuously while cycling. We actually took the battery off. We kept it in a holder, took it off the beam line, cycled it, and did some other work. So we didn't want to waste the X-rays and put it back in. Our system is such that it's very repeatable. If you go back to the same position, you'll find your cell. And the human eye is really good at picking out particles. So we actually took overview scans of the area and found some distinctive markers. And it was very easy for us to find that sample again. We were actually really worried about it, but it ended up being not a problem at all.
Good, good. I hope it answers your, yeah. Go ahead, Johanna. Yeah.
I would only add that for heating studies, non-battery related, like catalysis studies, there we have to track the particles while we're heating, while we're ramping up to temperatures, just because everything's thermally expanding. So that's the one time that we actually track particles because everything's moving.
Good, good. I hope, Gustavo, it answers your question. Okay. Then we have lots of questions from Anshuman. Anshuman, can you please unmute yourself and go ahead? That will be better, I believe. Okay. Yeah. He asks, I think you can check the chat also, Johanna.
Yeah.
Anshuman, you can also unmute yourself and go ahead if you want. Yeah, it's question 3. When you're with 2 alloying elements.
So will tin antimony with 2 alloying elements have a greater volume change compared to tin or antimony intermetallic with non-alloying elements, given the same porosity? Yes, because they both contribute to the volume change. I think I'm understanding this correctly. So for example, if you had tin copper and the copper is not going to alloy, it's just going to be structurally there, then you would have less volume change. There's also, we're doing some follow-up work on the tin antimony stuff to see, obviously it's not really an alloy anymore, it's an intermetallic, so how much aggregation of the tin and the antimony with their own pure metals is happening as they alloy and de-alloy with lithium. So we're trying to figure out the best way to track that over time, but that's a tricky question.
Yeah. And he has one more follow-up question about lithiation.
Is the lithiation of lithium cobalt oxide forming cobalt particles reversible? We do have data on that, and I think we have data on pushing it all the way to what 2.5 lithium, and then retrieving that. It was somewhat reversible, but it was forming other cobalt species which we couldn't identify, so we never published that data. I think if looking at the data we now have published, if you maybe only put in a lithium, so over-lithiated it a little bit, not fully to 3 lithiums, then I think it would be reversible as long as you avoid that cracking.
Understood.
Or nearly reversible. There might be some more side reactions.
Got it. So Rajesh has another question. Rajesh, you can unmute yourself and go ahead if you can, or if not, Can we use Electrochemical X-ray to observe lithium plating?
Plating stripping mechanisms on lithium electrodes on symmetric cells to understand the volume expansion contraction issues during formation deformation of SEI on lithium. So detecting lithium with X-rays is tricky. It's the lightest metal. So I have not personally done this, but I've seen some really beautiful work by a collaborator using micron tomography, so not nanotomography that I showed, micron tomography to see if you could see lithium metal on lithium. And you can see that in sort of a contrived cell. We are actually looking at zinc batteries because zinc's a lot easier to see. So we're looking and exploring zinc stripping and plating. SEI is also very difficult to probe with X-rays. It's all low Z materials. It's also very radiation sensitive sometimes if you're looking at it with the electrolyte around. So there's been some really good work on soft X-ray spectroscopy of SEI growth on, I think, copper originally, but I think they're working towards doing it on lithium as well.
Good. And I had one question. Is there a possibility to detect core temperature using X-ray, this technique, core temperature of batteries? Right now, let's say
so like using it as a thermal probe?
Yeah. At the core.
So, yeah, so we've some collaborators of mine have some work using X-ray diffraction to look at, for example, the copper current collector, and if the temperature increases, the Bragg peak will shift in the XRD. They're exploring that as a possibility. I don't know how big the shift is going to be, how sensitive it will be, and it's an average measurement, so you'd average through the battery. So whether you could distinguish that from the core from the outside, I'm not sure.
Yeah, because there have been.
I think it's.
Yeah. Go ahead. Complete the thought. Yeah.
I think putting thermal probes inside batteries is probably easier. And more accurate.
Because I've seen some work where they have tried to do constant temperature, constant current charging to improve the performance. Yeah, instead of just constant current, constant voltage. Yeah. So one more question by Tugba. I hope I pronounced it correctly. Have you ever tried this method in Operando for lithium sulfur batteries? Yeah.
So I have some published work on lithium sulfur batteries. For TXM, we can see the sulfur dissolve into the electrolyte and whether or not it reforms. We can't reach the sulfur edge for the chemistry imaging on the transmission X-ray microscope, but we do have a scanning probe, which goes to about 1 micron, 2 micron resolution. And there we have done some in situ work at a cross-sectional cell, looking at where is the sulfur species, the polysulfide species in the electrolyte. Are they on the lithium side? Are they staying on the cathode? Things like that.
Got it. Yeah. Then we.
So if you email me, I can send some.
Yes. Please email Dr. Johanna.
Email address is up there.
Yeah. And also thank you for uploading all the videos. I saw for your 2014 paper, you had uploaded the videos as well as supplementary material. It was also pretty cool to, yeah. In your references, the first paper. Yeah. So we have one more question from Anna. Yeah, Anna, can you unmute yourself and go ahead if possible? It's question 7.
I can. Can you hear me?
Yes,
yeah,
yes.
Hi.
Hello, Johanna. It's nice to.
Hi.
Well, it's not nice to see you because we're very far from each other. We are here in Brazil. But very nice talk. And I have a question. I think it's okay. It's not specific to perovskites and capacitors, but I think it has to do with perovskites. Can you use this X-ray microscopy in order to track the degradation in perovskites? And also there are a lot of work, I think, people really want to ask what's going on the interfaces with perovskites and other materials in a solar cell. I mean, the interface with perovskites, with the whole transport material, or even with electron transport material that sometimes can be an oxide, or for instance, a polymer or organic molecule. So can you use these techniques, both the transmission and also the mapping, to actually track what's going on there?
Yes, assuming that there is a heterogeneity. So as long as they're not uniform films where everything's happening simultaneously, because then it'll be a very boring image. But yeah, you should be able to track it, track the chemistry, 2D mapping. 3D is going to be harder depending on the size of the object. So we like cylinders because they rotate nicely. A flat film is a lot harder. Our chemical resolution in terms of the detection limit, we'd struggle to see something thinner than probably 100 nanometers, maybe 50 nanometers of pure material. But the easiest way is to just put it in and see. We did some perovskite work of unannealed samples with Mike Toney, and we found that the X-ray beam just made the chlorine come out. So it was pretty much impossible to look at these materials without basically annealing them with the X-ray beam. But the annealed samples should be fine.
Okay, good. I think I have a work for us to do together. I'll keep in touch.
Yeah, let me know.
It's a follow-up. Yeah, it's a follow-up actually work from a paper that we published in Nano Letters this year, and something is going on actually on with these 2D perovskites, and you can see by the in situ GWalks, but I think it's going to be important to have more information about the composition.
Yeah, send me an email. We'll chat.
Yeah, I do it.
Yeah. Thank you, Anna. Thank you for your question. I think Murad, you can go ahead if you want. Unmute yourself and go ahead. Last question. He asked, is it possible to detect hazardous gases during thermal runaway of cells by this technique?
So we don't really detect, most gases we're not sensitive to. For the catalyst work, we have hooked up a mass spec on sort of the out valve to track what materials we're forming. We could think about doing something similar for a battery. So to look at the morphology changes, but then map that to whatever you're seeing in that spec. But no, the X-rays aren't going to detect the hazardous gases.
Yeah, I think, yes. Thank you, Johanna. You answered all the questions. Thank you very much. Thank you for your time. Yes. Thank you for your time. We greatly appreciate it. And that too early in the morning, we picked your brain. And thank you very much. Please email Dr. Johanna. She'll be more than happy to answer the question. And please go through the published papers as well, as some of the work has been covered there as well. So
yeah, and if anybody needs any PDFs, just let me know.
Yeah, sure. So thank you, and we had audience from all across the planet, Johanna. So thank you very much for your time. We greatly appreciate it.
Okay.
Yeah.
Thank you.